The cell cycle must be very carefully regulated. And when it's not carefully regulated, then there can be things such as tumors that do result from that. So in this slide, we're seeing an example of normal cells and how they form a nice pattern. We have nice layers of cells. And then we also have a contrasting picture where we have, in the middle, we have a tumor. And that tumor forms because there are certain cells that do not have carefully regulated cell division. So what that means, if we just look at this right here, is that the cells divide uncontrollably. So even when we don't need new cells produced, those cells are actually dividing just again and again and again. So they're not responding to the normal control mechanisms that are in place. And unfortunately, this can cause a tumor, and in many cases, that tumor can be cancerous. So when we talk about cancer, the main problem with cancer is that you have cells that do not respond to the normal control mechanisms that are in place for cell division. So clearly controlling cell division is very important, and we have a number of molecules that are involved in controlling that cell division. So we have both positive regulators, and positive regulators would stimulate cell division, so they would cause it to take place. And then we also have negative regulators that cause negative regulation of cell division. So when we talk about getting tumor formation or cancer occurring, the problem is typically an error in the positive regulation system. So what generally happens is that a gene that controls a positive regulator is mutated. So this means that the DNA is changed in some way. And the DNA could be changed in response to a number of things. Changes in DNA could be a result of the environment. So if we're talking about something like UV light, that can cause changes in the DNA. It could also be something like a chemical that causes the changes. We know that there are chemicals that are called carcinogens that can mutate our DNA. Or there's also cases where it's viruses. So you could have a virus that causes a change in the DNA. Okay, so all different ways that we can actually end up with changes in the DNA, which we would then call mutations. But the end result is that you have a gene that is supposed to positively regulate the cell cycle, but somehow it's not correct. And that means that it just stimulates the cell cycle to take place and go round and round really all the time when we don't have a real need to produce new cells. We do have a special term for genes that are involved in positive regulation of the cell cycle, and that is a proto-oncogene. So with proto-oncogenes, they are involved in just normal regulation of the cell cycle, but if they do become mutated, then they can potentially cause cancer. If mutated, then we can have them cause cancer. And so then we call them, if they are actually resulting in cancer, we call them oncogenes. So a properly functioning proto-oncogene is not a problem at all. It's actually a good thing. It helps us to have proper regulation of our cell cycle. But if somehow it is mutated, and mutations are really very random, if it is mutated, then potentially it could cause cancer. And in that case, we would call it the oncogene. So these genes are going to code for negative regulators of the cell cycle. And that means that they actually prevent uncontrolled cell division. So this is very important because they will help to keep everything in check. And when we don't need cells to divide, they will prevent that from happening. However, if these do get mutated, that could also cause a problem because when things are mutated, many times that means they're not functioning correctly. So if you had a tumor suppressor gene that wasn't working, you might actually get that uncontrolled cell division, which could then lead to a tumor and it could potentially be a cancerous tumor.